Hello, everybody, and welcome back to our General Biology 1 series. Today we're going to be talking about photosynthesis, and in a place like New York and Manhattan especially, photosynthesis is probably not happening in a lot of places. Um, but Central Park would definitely be one of them. So that's what's shown here in this picture. Now, Central Park, of course, is a small urban park. It's actually pretty good size, but it's an urban park in the middle of the concrete jungle here. And one thing that's noticeable is when you go from sort of the streets of New York into the parkland of Central Park, you often notice a change in temperature, specifically being a little bit cooler. So, cooler temperatures in the forest of Central Park compared to the surrounding concrete jungle. Why do you think that is? Well, certainly New York is probably the prime example of this in the United States. It's the most urbanized landscape in the country. But all cities are struggling with something called the urban heat island effect. And this effect of parkland is very noticeable. When you transition from downtown to a more suburban area to areas where there's parkland, you notice that the afternoon temperatures and temperatures in general are a lot cooler in green spaces. So there's been this push towards maximizing urban heat island mitigation by just simply relying on trees, the stuff that was there beforehand. But why is this happening? Well, in the short answer, chemistry, and specifically the chemistry of water. Parks, with their plants and their trees, they're undergoing photosynthesis. And photosynthesis, if you recall, you're taking carbon dioxide out of the atmosphere, mixing it with a little bit of water, and you are creating sugar and oxygen as a byproduct. But you're also generating water, right? And that water ends up getting released in terms of evapotranspiration. And you already know that water has a very unique property in that it has a very high capacity for it to absorb heat, right? A high specific heat capacity, in other words. And because of that, it's able to take away heat and push it back out into the atmosphere and get it away from the ground. Whereas in a downtown area, that heat is sticking around, right? It's not as humid. So it's the humidity of the parkland that actually helps to get rid of heat. And it produces a sort of a heat sink that then dissipates into the atmosphere, right? And when you combine that with all the other effects, such as removing pollutants and just providing shade, you could see how a city would do really well to put as much park as possible. And so I like to be very applicable with the biology knowledge that I like to spill, but chemistry is playing a role here. So it's a perfect example of a big landscape being affected by a small biological process and the effect it has through some sort of little chemical idiosyncrasy of water being able to pull off high specific heat capacity, which, if you recall, has to do with those hydrogen bonds. So cool stuff. Now, photosynthesis. You know plants do photosynthesis. You know they do it through these chloroplasts, and inside these chloroplasts you have these thylakoids and then chlorophyll, right? So it's the chemistry of chlorophyll that we talked about previously. Now we're going to talk about the actual process of photosynthesis that involves chlorophyll. So chlorophyll is not just floating around, it's actually part of a bigger system, a photosystem. So a photosystem is basically a large protein complex that incorporates not only chlorophyll but other types of pigments as well, like carotenoids. And it's basically built around something called a reaction center. And the whole thing together, the whole photosystem is embedded in the membrane of the thylakoid. Right? So if you recall, the thylakoids are those pancakes, the membranes of which will kind of be the site where photosynthesis is going to start, right? And the whole thing behind a photosystem, as the name suggests, it has to do with photons, right? You're basically producing this specialized structure that's going to take light energy when a photon hits it and turn it into something chemical. So as this is the case, you have chlorophyll all over the place, but you also have these specialized chlorophyll molecules. And long story short, this is a redox reaction center. So beyond just being a reaction center, it's a redox reaction center. Because what you're going to do is you're going to take the photon hitting a pigment, with all of that energy inside of the photon, and you're going to use that energy to drive forward a redox reaction. And it's going to center on the reaction center, which is the middle part of the light harvesting complex. And in there, you have a special set of chlorophyll molecules. And what they're going to do is they're going to create, they're going to get so energized from all of the energy in the system that they're going to go ahead and get oxidized and lose that electron. And that's going to be accepted by our first thing to get reduced, our primary electron acceptor. Now, what type of reaction this is, I already told you that it's a redox reaction because it involves the transfer of an electron from one molecule to another. But where are the electrons coming from? Well, we'll get to that. But long story short, it involves water, right? So this is a photosystem. There's a couple of them. We'll get to it. So you have a photosystem 1 and you have a photosystem 2. And what differentiates the two is the wavelengths that they're absorbing at, right? So the photons are coming in. If they're coming in at a particular wavelength, one of the photosystems is going to react to that wavelength more than the other. And the reason is because of the specialized chlorophyll A molecules that are designed basically to have the absorbance at that particular peak. So you have a P700 chlorophyll A molecule, and you have a P680 chlorophyll molecule, right? So those are just the nanometer wavelengths that they're responding to. Now, in a plant, you have in the thylakoid membrane two photosystems that work together: photosystem 1 and photosystem 2. Now, in order to make things as complicated as possible, the first one that works in the pathway of photosynthesis is actually called photosystem 2. And it's the second one that's called photosystem 1. And the reason is not because they're named on their basis of where they are in the pathway, but they're named on the basis of which one got discovered first, right? So it's kind of a stupid thing. I feel like they could just switch this very easily and call them something else, but whatever. So important note, photosystem 2 is the first photosystem as you're making, as you're going through photosynthesis. Photosystem 1 is that second one. Now, what links these two together is a series of proteins and integral proteins that are embedded in the membrane that are also enzymes that are also capable of doing redox. And together they form what's called the electron transport chain. And it's the electron transport chain that's going to create the ATP, create the chemical energy that's going to as the electrons move through them, it's going to create the ATP. And that ATP is what's going to power the rest of photosynthesis. So you can see here in the picture, photosystem 2, and you have water. Notice that water is going into this system at that P680. What it's doing is it's actually donating its electrons. Every time you have two water molecules together, well, you donate those electrons. You can follow them with the hydrogens, right? But the electrons are being donated, right? So they're being oxidized, and as H2O gets oxidized, two of those H2O molecules, those free oxygens, they're going to bind up, and as they bind up, they become O2, right? So that's where the oxygen's coming from. So right off the bat, the photon of light is hitting the reaction center, or the photon of light is hitting the photosystem, and you have water being used right off the bat to donate its electrons to the system. Now, as that electron gets moved from the P680, it's going to be sent up into the primary electron acceptor and then move down the chain, the electron transport chain, and then eventually get to a point where it's going to be re-energized in photosystem 1. The whole process will take place again, except this time you don't need water because you already have the electrons moving through the system. And then that's going to be used to power the last bit, which is to finally end up with a molecule that's going to produce the NADPH that's necessary for the next phase of photosynthesis. So that's a quick, in a nutshell kind of thing, but we're going to get into more detail because that's what we do. Now, this whole thing is often referred to as non-cyclic electron flow, and we use this idea of a Z-scheme. So, it's basically take a Z, move it on its side, and that's basically what you're looking at. And the idea is that you're following the 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 thing of energy, right? So starting at the bottom of photosystem 2, you have very low energy, but as light hits it, the electron is moving up. So it's got a high amount of energy. And then as it moves through the transport chain, it decreases, right? The energy that's inherent in that electron decreases until it gets to photosystem 1, gets re-energized for that final step. So it has sort of a Z pattern. So if I ever use the term Z scheme, or you hear the term non-cyclic electron flow, that's what we're referring to it, right? So what is NADPH? Do you remember what it is? What its role is? Okay, so NADPH, right? That's that special electron shuttle, right? It's a high energy product, and it's called NADPH because it's in its reduced form, right? So NADPH is the reduced form. When it loses that electron, it also loses that hydrogen, and it becomes NADP plus. And NADP plus is the oxidized form, right? So what is the purpose of NADPH or NADP plus, which is, two sides of a coin, right? Well, the reason you have this thing is, like the name suggests, you're moving electrons, right? It's a high energy product that is going to have electrons that it's ready to give. And you'll learn later on that, this is all happening in the light reaction, but later on, after the products of the light reaction are going to be shuttled into the Calvin cycle, or the light or the dark reaction, or the light independent reaction. And there, NADPH will then donate its electrons. So it's there to basically start a whole redox reaction in a whole other part of the plant. Right? So it's kind of cool, right? It's basically taking electrons, shuttling them to the next spot where they're then going to start a redox reaction over there. But we're getting ahead of ourselves. But we will get there eventually. So another way to think of that Z scheme, or that non-cyclic electron flow, is, with this analogy with these workers, right? Their goal is to get the electrons into the bucket, and that bucket is NADPH, right? So NADP plus is what that empty bucket would be. As soon as it gets the electron, it becomes reduced, right? Because it's gained an electron, and it becomes that NADPH. So a photon is the energy required to bounce that electron up there. It then rolls through, and you can use the gravity as it moves down to power a mill that produces ATP, and then it gets there and needs to get hit again with another photon at the photosystem 1, and then gives it just that right amount of energy to get it in the bucket, right? So can't help but feel like it'd be a lot easier to just maybe put the bucket on the ground, but that's okay. We're just talking about an analogy here. So this is all happening in the thylakoid membranes, right? So you have the photosystem 2, light hitting it, water donating electrons, becoming O2 in the process. That electron is then going to move through the electron transport chain, which we basically think of three elements to it. There's a plastoquinone, the PQ. That's the first electron acceptor of the electron transport chain. It then gives its electron to the cytochrome complex, which is, as the name suggests, it's very complex. And then that electron will then move to the last part, which is the plastocyanin. Then gets into photosystem 1, light hits it, that electron moves up through there, ends up going into a thing called ferredoxin, or FD, and FD is then going to give it to an enzyme, an NADP plus reductase, which, as the name suggests, reduces NADP plus to NADPH, right? And now that electron is safely at the other end, ready for the Calvin cycle. So we already talked about how NADPH is necessary for the Calvin cycle, or that dark reaction. But what about the ATP, which is the other high energy product? Well, this one's a little trickier, but at the same time, I feel like it's pretty intuitive. As the electron moves through the electron transport chain, there is actually a pump. And what that pump does is it uses the energy from the electron moving through to power protons being brought into the thylakoid, right? So again, if you're looking at protons, so we're talking about H plus here, there's low amount concentrations of H plus outside of the thylakoid in the stroma. There's high amounts of H plus inside the thylakoid, in the thylakoid space, right? So low outside, high inside. Well, if you're trying to bring in protons from outside into the inside, well, you're going from an area of low concentration to an area of high concentration. You can't just do that. You need energy, right? So that energy is coming in the form of that electron moving through. So on top of everything else, that electron is powering a pump, moving protons into the interior. So now you've got protons being pumped in from outside. You also have protons that are being produced as water is being oxidized, losing its electron, but it also frees up these protons. So you have a high concentration of protons out there, right? So now you got this difference across the thylakoid membrane in the concentration of protons. What is that? Well, that is energy, right? That is energy that can be used to produce something, right? So this process here is basically altogether called photophosphorylation. And the reason it's called that is because by creating this difference in H plus across the membrane, well, if you were to somehow open up a channel, then the protons would do what they naturally do, which is they would leave from the area of high concentration inside the thylakoid to the outside stroma. And they would do that on their own free will, and they would have energy while they're doing that. If you couple this to an enzyme like ATP synthase, you can use that energy to photophosphorylate ADP and an inorganic phosphate into ATP. And that's the next part, right? So we showed you earlier pictures where there was the ATP just being made magically. This is the actual process of that. The bottom line is you'll end up with producing large amounts of ATP. You're also producing large amounts of NADPH. You've already produced oxygen as your byproduct. So we're starting to get all the products of the light reaction. It's also convenient that the ATP and the NADPH is being produced in the stroma, which is where the Calvin cycle is going to use them. So pretty cool. Now think about the difference between the stroma and the thylakoid, right? This is a kind of basically the same sort of schematic, right? So again, I just kind of want to emphasize this because I think it's pretty cool. Water is being split, you're producing protons. You're also using the electrons to power a pump that's going to move more protons from the outside into the interior of the thylakoid. And then as things move through, those protons are being pumped out, right? And as they're being pumped out, you're using that, or not pumped out, sorry, they're just moving out on their own. You're using that energy to power the production of ATP through ATP synthase, right? Now, a lot of those protons that get moved out, well, a lot of them come back in. Some of them are there to be taken up by NADPH or NADP plus to turn into NADPH, right? So everything's kind of in a nice little balance here, which I like, right? So once again, I tell you this all the time. Everything that happens to you, everything that happens in life, it's because something either changed its shape or something changed its charge, right? And in this case, it's the charge differences between the inside and outside of a membrane that is powering the ability for plants to produce their own food, right? So this whole process is also an example of what's called chemiosmosis, where you're coupling the production of ATP with the movement of an ion, in this case a proton. I want to remind you, I know you're learning about this, and you're learning about photosynthesis, and you're learning about the chloroplast and how they're doing it. And in a chloroplast, the high pH environment is the interior of the thylakoids, right? So it's on the inner membranes. In the case of mitochondria, the same sort of process is happening, where you're coupling the electron transport chain with ATP synthase and production. And you're using this sort of pump to produce something that's going to push through and produce ATP. So by generating a gradient, you're doing the same thing. Now in the case of mitochondria, they're almost like the flip side, right? So in a lot of ways, the chloroplast and the mitochondria are kind of opposites of each other in how they work, but they have the same mechanisms at play. In this case, using a proton pump to create a electrochemical gradient across a membrane that you can then use to power the production of ATP. All right, so regardless of which organelle you're looking at, whether you're looking at aerobic respiration in the case of mitochondria or photosynthesis in the case of chloroplasts, it's the same sort of process. So understanding one will allow you to understand the other. Okay, so let's say you take a plant cell and you illuminate it. Which of these five will have the lowest pH? Hopefully you're illuminated enough to guess E, the thylakoid space, right? So again, that's where you have the highest concentration of protons. So high H plus concentration means a low pH. So it's a very acidic environment, in other words. All right, so now we've talked about photosynthesis as pertaining to the light reaction, right? So the light reaction is happening in light, and you're producing the products that you need to power things through, right? You're producing ATP, you're producing NADPH. So now we have what it takes to do the Calvin cycle. So where's the Calvin cycle happening? Well, it's happening in the stroma. What does it need to proceed? We already mentioned it needs ATP, it needs NADPH, right? So NADPH is bringing that electron that we need, ATP is bringing energy. So what's going to be the product of the Calvin cycle? Well, if you recall, the product of the Calvin cycle, the end goal is to produce a sugar, right? Glucose, although it's not really glucose that gets produced right away. So we'll talk about that. But the Calvin cycle, as the name suggests, is a cycle, and you need to start somewhere. So we're going to start with a molecule called ribulose biphosphate, or RUBP. And RUBP is a sugar. It's a 5-carbon sugar, and it's going to be used to fix the carbon dioxide that's in the atmosphere. And it's going to do this with the help of rubisco. And rubisco is a very, very important enzyme. And the name is cool, rubisco. And so let's talk about the first step, carbon fixation. As I mentioned, rubisco is the key enzyme here. Rubisco is thought to be the most abundant enzyme on this planet. If you think about it, every cell in every plant that does photosynthesis will have rubisco. It's what they call a constitutive enzyme, meaning that they produce a ton of it all the time. It's not something that gets produced at a certain time or under certain orders from, say, gene switching on or something like that. It's just made all the time at high concentrations. So it could very well be the most abundant enzyme on the planet, which is pretty cool. So the first step, as I mentioned, is taking that carbon atom and attaching it to your RUBP. So you have RUBP coming into the system. So in this picture here, it shows the molecules. Each of those gray dots is a carbon atom, right? So of course there's going to be other things coming off of it, but we're just focusing on the carbon today. You'll also see the small p in the circle. Those are the phosphates, and they come and go depending on the reaction. The other thing I'll also mention is you have a little number right by the molecule in each of these steps. That's the number of molecules that are produced, right? or the number of molecules there are. Now, in order for this particular diagram to make sense, you have to basically go through it 3 times. And by going through it 3 times, that means each time you're using a carbon dioxide molecule. And you need to do it 3 times in order to produce a single glucose and to regenerate the whole system. Now, in the reality, this thing's just going on and on and on, right? It's like a turning wheel, just all day long, all day long. So, we do these things so we can understand them graphically. But just remember that it's less of a picture and more of a video. It's just a constant video on loop of this thing turning around. So what happens? You enter in carbon dioxides, you do it one at a time into the picture. It interacts with Rubisco and RUBP. And what happens is, as you can see, it goes from having 5 carbons, RUBP does, to now having a 6th carbon coming off of that CO2. So you've just done it. You've taken a floating gas and turned it into a real molecule, right? Or not that the gas is not a real molecule, but it's not something that's necessarily bioavailable to plants. So now you have this little intermediate, but what's going to happen very quickly is it's going to get cut in half. So you go from having 3 of these short-lived intermediates, 1 with each CO2, to now being cut in half to produce something called 3-phosphoglycerate. And the names are not necessarily important. Sometimes I call it 3-PG. And 3-PG, you have 6 of them because you started with 3 of those molecules and you cut them in half. So now you have 6 of these things, each with a phosphate group. And that's it. That's the first step. That's the carbon fixation step of the Calvin cycle or the dark reaction. So what happens next? Well, what happens next is you have to reduce this molecule, right? So first off, you're going to attach phosphate groups to it. So it shows here at the 3-PG mark up there in that first phase. You're now entering the second phase of the reduction, but before we get to that, 3-PG has another phosphate group attached to it. So you see that you went from 6 3-PG to now producing 6 of something called 1,3-biphosphoglycerate. So biphospho, 2 phosphates, right? And the 1 and 3 just tells you which carbon they're attached to. So attached to the 1, attached to the 3rd carbon, there are phosphate groups attached to both of those. This is also very short-lived, right? You use 6 ATPs to do this because you have 6 molecules to do it with. Then what's going to happen is this biphosphoglycerate is going to be reduced. And it's going to be reduced because you have these NADPHs that you got from the light reaction. And those NADPHs have electrons attached to them. So what they're going to do is they're going to basically reduce your molecule to something called G3P, glyceraldehyde 3-phosphate. Now, superficially on this picture, it looks the same as a 3-PG molecule because it basically lost a phosphate group. Okay. You can't really tell because what's happening is electrons have been donated, protons have also attached to this molecule, but you don't see any of those because all you're seeing is the carbon skeleton. Right? So that's something to bear in mind. Stuff is happening here. So how many electrons were needed to get these? Well, to produce this right here to this stage, you're going to need 6 electrons. 6 NADPHs means 6 electrons have been donated, right? So now you have G3P, you have 6 of these molecules. Notice you have this little offshoot, right? One of these G3Ps is leaving the system. So now you're left with 5 G3Ps. And one of these G3Ps is going to be used to make glucose. And it's going to do this through this other process, right? So G3P, one of those is being produced. So again, 3 carbon dioxides entered this Calvin cycle. 1 G3P molecule is leaving the cycle. So how many G3P molecules do you need to make a molecule of glucose? Well, if you did your math right, you know that G3P has 3 carbons, and you know that glucose is an example of a hexose sugar, meaning that it needs 6, right? So Greek, hexe means 6 in Greek. So you need 6 carbons to do so. So you would need to have at least 2 G3P molecules to produce a molecule of glucose. So you enter in a carbon dioxide, it goes through these steps. You enter in another carbon dioxide molecule, it goes through this step twice and produces the two G3P molecules that leave the system to produce glucose. But you still have one last thing to do. Well, the Calvin cycle is not finished because you still have to regenerate, right? So in order to regenerate, this is where that third carbon dioxide comes in. So in a regeneration, all that's happening is you're adding more phosphate groups, right? And in doing so, you're basically reconstructing that RUBP molecule. But in order to make the math work out, you have to add in a third carbon dioxide to do this. Once you do this, the whole thing can regenerate. All right. So G3P then gets phosphorylated again, and you produce, and then it combines again, and then you produce a 5-carbon sugar, right? That's where the third carbon dioxide is coming in. So in the end of the day, if you do the math, you'll have regenerated 3 molecules of RUBP from the 3 carbon dioxides you entered into the whole thing. All right? So again, the 3 steps: carbon fixation, reduction, regeneration. So you're producing all this sugar as a byproduct, right? One thing I do want to emphasize, sugar is energy. It's metabolic energy. That same sugar that's produced in the chloroplast will be used as the metabolism for the plant, right? Or stored somewhere for hard times. So metabolism is going to happen. You're going to produce glucose, and you produce other sugars that are 6 carbons, and you're going to produce dimer sugars like sucrose, and those will be the kind of things you're going to store. But eventually, in order to maintain yourself, that metabolism is going to require you to basically burn these sugars again. So you're almost going to flip side. Sometimes people confuse the Krebs cycle with the citric acid cycle, right? So the citric acid cycle is what's happening inside the mitochondria, and there what you're doing is you're regenerating CO2, and you're also using up O2 in anaerobic glycolysis. This happens in animal cells, but it's also happening in plant cells. They have mitochondria as well. So something to think about, right? That sugar is being used. Right? Okay, so let's take a step back and talk about where this stuff comes in a plant. So if you look at any plant, and you look at the leaves, and you look at the underside of the leaves, and especially if you have a microscopic eye or a little magnifying glass with a good high power, you can actually make out the little structures where gas is being exchanged in and out of the plant. And these are called the stomata. Right? And the stomata just means mouths, basically in Greek. And gas are going in, carbon dioxide is going in, oxygen is leaving through this thing, right? And any gas will go in and out of these things. They're basically just openings to the interior of the leaf. So what other sort of things might be going in and out of there? Well, the big one is water. So water can present a challenge, but before we get to that, I want to talk a little bit about how carbon dioxide works. So if carbon dioxide levels are really high, Rubisco, which is a very, very, very good enzyme at fixing carbon dioxide, it will fix it. And it will do so in the way we just talked about in the Calvin cycle, where CO2 will be fixed to RUBP. And because the first product of that, besides that short-lived metabolite, is a 3-carbon sugar, we call it the C3 pathway, right? So this is what most plants are doing all the time. They're taking CO2 and they're fixing it to RUBP with the help of Rubisco in what we call the C3 pathway. However, there are situations where CO2 concentrations can start to decrease and start to decline. And as that happens, while you're declining CO2 inside of a plant, you're also increasing oxygen, right? Because oxygen is the product of photosynthesis, right? The light reaction is producing oxygen all the time. Now, Rubisco is a very good enzyme, but it's not the most specific enzyme. And in situations where CO2, which is its preferred, what we call, ligand or preferred substrate, when it starts to decrease, it can start binding other things with lower affinity, right? And in this case, it could also bind up O2. because CO2 and O2 are not that different in terms of how Rubisco sees them. So in situations like that, O2 will be bound up by Rubisco, and the enzyme starts acting like an oxygenase. And what you end up doing is you end up taking the O2 molecule and fixing that to RUBP. And this whole situation is an example of what they call photorespiration, because it's like respiration because you're using oxygen, right? Just like we use oxygen for respiration. So fixing oxygen to RUBP produces a toxic byproduct that a plant cannot use. And if it builds up, it could become a big problem, right? So this toxic waste product is building up and it's destroying the tissues and the plant, but at the same time, you're also not doing photosynthesis because you don't have CO2. So this is an issue, and this can arise specifically in the C3 pathway during a particular time. And under normal circumstances, it's not an issue. But the issue does arise when you have to deal with the stomata closing. So plants get their oxygen through the openings in their leaves called stomata. I just gave you that one, right? But in a situation of water stress, one way to avoid losing all your water, what a plant will do is it'll close its stomata. Well, that's a great short-term solution. But when you close stomata to preserve water, because water vapor will leave, right? We talked about evapotranspiration. That's what evapotranspiration is. It's the water vapor leaving through the stomata of the leaves into the atmosphere. That's great if you're Central Park trying to cool down temperatures in the middle of New York City. But for most plants, that can produce this issue because when you close a stomata, what happens to your CO2 concentration inside your plant? Well, it's still getting used, right? So internal CO2 concentration is going to decrease. While that happens, O2 is increasing because whatever CO2 is in there is being used for photosynthesis, the light reaction, you're producing O2. So you're building up this problem. So, we often say that plants walk a fine line between hunger and thirst because they're hungry, they're using up CO2, and they're needing CO2 to come into the system, but while they're doing that, they lose water, right? So it's this eternal struggle between, okay, what can we do? We need the CO2 to eat food, but we also lose water when we do it, right? So it's this constant struggle between hunger and thirst for a plant. So what can plants do? Well, for a lot of plants, it's not an issue. They use the C3 pathway and they're fine. But there are situations where some sort of adaptation is necessary. So again, this is just an overall sort of view of what's happening within a plant. Notice, all of this is happening inside of a single cell. In this case, it's happening inside of the chloroplasts, right? So every cell with chloroplasts, plant cells, are doing this, right? So the light reaction, Calvin cycle, that's happening inside of a typical C3 plant. But, well, one thing you can do is maybe separate it. So what you do is you basically set up a situation where you can't let Rubisco interact with oxygen. So you separate them out through some kind of spatial separation. A lot of commercial plants do this. A lot of grasses. Most grasses are doing what they call the C4 strategy, right? Sugarcane is a fancy type of grass. Corn is a fancy type of grass. Wheat is a fancy type of grass. Most of our agriculturally important crops are grasses, just so you know, right? You cut them at the top, they still keep growing because they grow at the base. What they do is they basically set up a situation where they fix carbon dioxide in one cell, but then they do the rest of their Calvin cycle in another cell. So what do? CO2 comes in, they open their stomata, CO2 is coming in, and it gets immediately bound up in mesophyll cells by a new enzyme this time, PEP carboxylase. PEP carboxylase, or Pepsi as I like to call it because I like to drink Pepsi. Not related though, even though they both have carbon. Pepsi is actually named for pepsin, the digestive enzyme. But CO2 is bound up right away at the site by this new enzyme, PEP carboxylase. And it's very, very competitive. It's actually more competitive than Rubisco, meaning that it's going to bind up CO2 way quicker than Rubisco does. So it outcompetes it. And when it does so, it produces a molecule that isn't a 3-carbon molecule like in the C3 pathway, but a 4-carbon molecule, hence the name C4 pathway. So this 4-carbon molecule or compound is then taken out of the mesophyll cells and moved to a new set of cells, the bundle sheath cells. And at the bundle sheath cells, well, then what happens is there's an enzyme that breaks down that 4C compound and re-releases that CO2. So what does that mean? So now it means you can fix carbon dioxide in the mesophyll cells and then re-release it in a whole new set of cells, the bundle sheath cells. And while you're doing this, you're basically confining the Calvin cycle to those bundle sheath cells and nowhere near the stomata. So you're not going to suffer that situation where oxygen buildup is going to throw off your system. So how does this look? Well, this is what it looks like. So you have your mesophyll cells, which are closer to the stomata. You could see in this diagram here, the CO2 coming in. It's coming in through the stomata. The first type of cells it's going to interact with are those mesophyll cells. PEP carboxylase is quickly going to take it and turn it into a 4-carbon oxaloacetate, oxalic acid type thing, right? It doesn't matter what it is. But it's all happening in the mesophyll cells. Then what's happening? Well, it just gets imported through special channels into the bundle sheath cells, which you see are located, is the lower cell there. And in the bundle sheath cell, it's going to re-release that CO2. It's going to release this CO2 from this 4-carbon molecule. It becomes a 3-carbon pyruvate at the same time, and then that gets cycled back in. So they basically have 2 cycles. Now you have the CO2 released in the bundle sheath cells, and it's going to go through the Calvin cycle as before, produce a sugar, and that sugar will eventually make its way into the phloem. That sugar, which will become eventually a sucrose, it's going to make its way into the vascular tissue and get sent all over the place. Vascular tissue, by the way, phloem is what we're talking about. Spoiler alert. So again, you're separating out the stomata from where the Calvin cycle is happening. So the O2 buildup is going to happen somewhere else. Light reaction is happening in mesophyll cells, but Calvin cycle is happening in the bundle sheath cells. So it's a spatial separation, right? They're still dependent on each other. They're just happening in different cells. Now, if you have a situation where you're in a really tough spot, where you really have to be concerned about your water, you could operate something called the CAM strategy. So we think of the CAM strategy, I always think of things like agaves and aloe veras, cacti, pineapples are also a type of thing. And in these, because they're accustomed to very hot, dry, arid environments, they basically have a situation where they can't really open their stomata during the day, right? Because they will quickly lose water. In a C4 plant, they can do that. They can do it during the day. They just do it in different cells. But in their case, instead of separating out by cells in a spatial sort of way, they do it by separating out in terms of time or temporal separation. So during the day, that's the hottest part of a day is during the day when the sun is, especially out in the desert, right? So during that time, what they'll do is they'll close their stomata. And they're doing this because they want to conserve water and minimize evapotranspiration. But at night, they open their stomata, right? And they open their stomata, and the CO2 will rush in, and they'll go ahead and do what the C4 plants do. They use PEP carboxylase again, and they fix the carbon dioxide. But they fix the carbon dioxide in those mesophyll cells, and they'll doing this all at night, right? So all of this stuff is happening at night, right? And the stomata are open. Then during the day, the stomata close, but you have all these 4C compounds sitting there. Well, now you also have, during the day, you have light, right? Go figure. So now you have the light to power the light reaction. Because again, you can't do the light reaction at nighttime. You got to do it during the day, right? But you can do the Calvin cycle part at nighttime, right? But you can't really do the Calvin cycle part at nighttime because you would quickly exhaust all of those energy products you need, like ATP and NADPH. You need the light reaction to be happening. So what they'll do is they'll close the stomata during the day, and that's when the photosynthesis is happening. That's when light reaction is producing stuff, and that's when the Calvin cycle is happening, right? The CO2 is released from those 4C compounds during the day. So during the day, it's photosynthesis as normal. But at nighttime, that's when they bring in the CO2. So I always think of it like a warehouse. Like, a warehouse gets shipments at nighttime, right? That's when things come in. During the day, they might be closed. There's some warehouses out there, they don't have do much during the day. They do a lot of stuff at nighttime, right? Or shipments in a store or something like that. So again, example of spatial separation. This is especially important for plants that grow in arid environment. So in many ways, it's the same sort of product. It's the same sort of situation. It's just that one's separating it out by separating out the steps in terms of space, different cells. The other one's doing it in the sense of separating them out in time. Same strategy, slightly different purposes, but both of these are ways to combat photorespiration. So here's a question. This is one of the coolest grasslands out there. These are the grasslands at the base of the Himalayan mountains. And these are grasslands. These are some of the tallest grasses. They grow several meters high. Very cool. You can see this Asian elephant just walking through with people on their back. There could be tigers in here. You would never know, right? Pretty crazy. Pretty crazy that there's grasslands like this. Again, an environment that's very rare nowadays, unfortunately. But they're doing their best to preserve it. They're good examples in places like Royal Cheatwan Park and stuff like that. But anyway, moving on. So let's imagine you do an experiment, right? So you're doing an experiment of photosynthesis. You're conducting the experiment during the day, and you're basically using carbon dioxide that has been labeled with a radioactive marker or an isotope of carbon, right? It's not really radioactive. C14 is, but whatever. Um, C14 carbon labeled carbon dioxide to follow it. And during this experiment, you notice that it is used to produce oxaloacetic acid. So you have oxaloacetic acid that has these 14 labeled carbons attached to it. So what type of plant are we talking about? Well, the grassland should give it away, but it's a C4 plant, right? Because the fixation is happening during the day. If it was a CAM plant, fixation would be happening at night, right? So because it's happening during the day and you're getting this 4 carbon molecule, this oxaloacetic acid first, as it gets fixed, that means you're not dealing with a C3 plant, or else it would just be Rubisco attaching it to RUBP. So the correct answer here is A. Now, which of these processes is happening in a C3 plant in the chloroplast during a sunny day? Well, it's a C3 plant, like this hibiscus that's being shown in the picture here. And in a C3 plant, uh, during the sunny day, you can do your light reaction, right? And the product of your light reaction is going to be those high energy products, ATP and ADPH, right? You're producing both of those in a C3 plant during the day. Now, what about at night in a CAM plant? Well, you should know that you're not producing ATP or NADPH at night. You're not producing ATP at all at night, right, in a plant. So A and B are out, and thereby D is out. So what you are doing in a CAM plant at nighttime, however, is you're doing your Calvin cycle. You're producing glyceraldehyde 3-phosphate, G3P, which is the precursor sugar that will eventually become sucrose, right? So in a CAM plant at nighttime, that's when your Calvin cycle is happening. And cool CAM plant here, you have not only the saguaros in the background, but you also have these crazy trees. They're called boojum trees. They're in very rare parts of the Sonoran Desert, particularly in a place called the Pincanzo Desert, which is Baja, California. So they're not really found in the U.S., but you see them in Mexico, and they're one of the coolest things out there, I think. And they're an example of a CAM plant. Okay, so let's talk about photosynthesis. Photosynthesis, in the end of the day, it matters. It matters not only for the obvious reasons in that it powers all of life here and it powers the food chains, but there's also interest in tapping into the potential that an artificial photosynthesis can make. Photosynthesis is a metabolic process that fixes carbon dioxide. And we know about climate change and we know about CO2 being a major contributor to climate change, and a lot of that coming from fossil fuel burning, right? That's the story. So CO2 is going up with industrialization and has been going up and continues to go up as we do things, right? And again, CO2 is not just a product of the fossil fuel usage, and not just it's not just about cars and things like that, but our entire lifestyle is powered by fossil fuels, whether it's electrical generation, whether it's production of plastics, everything, right? So there's a lot of incentive to find ways to take CO2 out of the atmosphere and to produce some of these things that are necessary for our current industrial species to use, right? So to produce some sort of chemicals that can potentially add value, value added or marketable chemicals. So there's a lot of research into seeing how carbon dioxide can be fixed to produce things like fuels, like hydrogen fuels, ethanols, methanols, also produce things like sugars, right? Imagine we could take CO2 and produce sugar out of it, right? Not have to rely on sugar cane to produce sucrose, right? That'd be crazy. Or acetate, which has other industrial applications, or using it to produce the methane for plastics, right? It's all a matter of finding efficient ways to replicate photosynthesis. So perhaps one day we could have artificial trees like these artificial trees that they have in Singapore. There's a Garden of the Groves. They have some solar panels attached to them, but I think their main thing is like being outlets for like pollutants and stuff like that. But they, Singapore, so they made them look really nice, right? And they have this walkway that goes around them, pretty cool. So if we can find a way to produce some sort of photocatalysts that will allow us to fix carbon dioxide out of the atmosphere and produce chemicals as a result, that would be a huge, huge thing. And the person who figures that out could be the first trillionaire, right? Or multi-trillionaire potentially. So a lot of really cool stuff being done on the bioengineering front, but again, we're talking about reactions that plants have figured out how to do a long time ago. All right, that's all for now. I want you to take care of yourselves, and I'll see you next time.